personal reflection. This is a picture of Knut Schmidt-Nielsen that I found obviously on the net, but it was when he was about this age, he contacted us in Stockholm because he was visiting Stockholm together with his wife, because his wife was Swedish and they were visiting her parents, and he wanted to use our library. And so for several summers he came and used our library, and to say thank you for that, he gave a doctoral student course each time on how to write an article, a scientific article. And we were very grateful, and it was a wonderful opportunity for our doctoral students to meet this fantastic and extremely renowned person under these circumstances. And of course, he is an icon in this field of animal physiology, and he's, in a way, just as we see here, he is, many of his experiments are textbook knowledge now. He's come so far with his experimentation that it's incorporated in our general knowledge of this area. And he also wrote an interesting and very personal autobiography, The Camel's Nose, and in this I found a section where he tells us about how they at Duke had trouble keeping their sand rats alive. And note, he says, despite growing fat and looking healthy, many died unexpectedly. And what he found then was that actually they were suffering from diabetes. And he notes further down, as you see on the page, kept on a limited ration of calories, the rats stayed well. They were given plants of the goosefoot family, so maybe we should introduce that for humans as well. With free access to food, they developed diabetes, and this is a pattern strikingly similar to the development of the disease in diabetes-prone humans. And in a way, then, this brings us this aspect of energy balance in diabetes back to what I'm going to talk about today, the brown adipose tissue, and the aspect that it's a mammalian prerogative. Work from our department that I'll be talking about has been performed by doctoral students Klaas Karneheim, Valeria Golozubova and Helena Feldman, together with postdoc Anita Matthias and a project leader Irina Shabalina and my husband Jan Nedergaard. The bioinformatic data was performed by James Timmons when he was at Karolinska Institute, and from the University of Ancona we've collaborated on human data with Maria Cristina Zingaretti and her boss Saverio Cinti. We can see in Gary Larson drawing here how there was about 75 million years ago a change in our climate. So some of the dominating species maybe gave way to the other smaller species that were arising. And why they probably survived, we would like to suggest, is that they had brown adipose tissue with the uncoupling protein UCP1. Now, to understand the role of UCP1, I want to take you back for a moment to the endosymbiont hypothesis, where a proteobacterium invaded the primitive eukaryotic cell, and after it resided there, it needed, of course, still to be able to exchange a lot of components over the membrane of this now becoming organelle, the mitochondrion, with the cytoplasm of the eukaryotic cell. And in order to do this, a large number of proteins were developed, based originally, we believe, on an early transmembrane protein with 2 alpha helices that went through gene duplication to become a tripartite transmembrane protein, and many of these then went through further duplications to become the members of the mitochondrial carrier protein superfamily. And these all have characteristics that I've indicated of these 6 transmembrane helices, proline breakers at the ends of the helices, and this is characteristic of all the mitochondrial carrier proteins. And a rootless tree can be generated with the sequences of known carriers. There are another 30 that we don't know the function of yet. And this we can see here, these different relationships with the substrate carriers, ATP, ADP carrier, and so on. And over here there's an arm of transporters which I would say have no known function. But they have a nucleotide binding pocket up here, all of them, and Daniel Riquet, who found one of these from the sequence, termed them an uncoupling protein, but later regretted this and said that they really should be called unknown carrier proteins. So this is perhaps really what we should think UCP stands for. And these are the older UCPs, the UCP2, UCP3, and what we call proto-UCP1 in a table, designed by Saito here in 2008. He collected sequences from all these species and was able to see their relationships. And proto-UCP1, this has no relationship really functionally, certainly not with UCP1, the mammalian one. Proto-UCP1 was identified simply by what's called gene syntony. That is to say, in the genome, it's possible to identify the genes on either side of the gene of interest. And when doing that, one could see that there was a mitochondrial carrier protein between these two conserved genes, one on either side. But this protein bears little relationship whatsoever with UCP, the later UCP, and we don't know its function, as I don't think we do with UCP2 and UCP3. So these were all present in all these beasts, and I'm sure they're very important proteins, but this one got a special one. And it got a special one, the uncoupling protein, from the extant eutherians, the placental mammals. But note, and this I think is important, there's a very long stretch here, so that evolution was very extensive in this time period before we got the UCPs that we know from the eutherians, and it's very distinct from the marsupials, monotremes, fish, etc. And they do have, these UCP1s have special conserved sequences, the functions of which we don't know yet, but they may correlate with the unique function which we believe UCP1 has, which is the mediation of the adaptive thermogenesis. So these probably important proteins are not uncoupling, are not thermogenic, and they're found in all vertebrates. And then we have the unique, we believe, uncoupling and thermogenic UCP1 only found in these eutherians. So how does the presence of UCP1 affect mitochondrial function? In mitochondria that don't possess UCP1, we have the respiratory chain pumping out protons, make a gradient that can be used then to drive the synthesis of ATP, to take up different compounds, ions, substrates, and so on. And then there's some probable leak over the membrane. And in such mitochondria, that we can now study as brown fat mitochondria, but in UCP1 knockout animals, we can see then when we add a substrate, we have a very low rate of respiration, and this can be uncoupled by the artificial uncoupler FCCP, so we know that the mitochondria here are tightly coupled. In mitochondria that contain the UCP1, we believe then that we have a function which returns protons to the matrix, and we see here then a very large difference in isolated mitochondria. Substrate now causes immediate oxidation. So UCP1 in isolated mitochondria is innately uncoupled. We can add a nucleotide. I showed you the nucleotide binding pocket. This closes the action of the uncoupling protein, returning respiration to the low level, the coupled level, and then this can be also uncoupled by artificial uncouplers. So UCP1 is a member of the mitochondrial carrier protein family. It transports the equivalent of protons across the mitochondrial membrane. I say the equivalent because, remarkably, we don't actually know what is transported, but the conclusion is, nonetheless, that it's the equivalent of protons. And this means then that we don't make ATP and energy is released instead as heat. This is in isolated mitochondria. In the cells, and look how beautiful they are. Here we have a central nucleus, small fat droplets, and very dense mitochondria with very dense cristae. Doesn't it look nice? In these isolated cells, of course, the UCP1 can't be active all the time. We have to have a regulation of its function. And the regulation then is from cytosolic nucleotides. So while we use a GDP in experiments, we think it's ATP, which normally closes the hole in the UCP1. And so if we're going to get any heat production in the isolated cells, we have to postulate that they're activators of this process. And already some 45 years ago, long before we knew of the existence of UCP1, we did suggest that this could be fatty acids because of the process of lipolysis. But this has always been rather contentious because in any mitochondria, fatty acids can uncouple. That is to say, they can introduce a leak. We see here in the UCP1 knockout mitochondria, we start adding fatty acids and respiration increases just as it did when I showed you the artificial uncoupler. So fatty acids can uncouple even in the absence of UCP1. What happens then, what's the comparison then if we have fatty acids added to mitochondria that contain UCP1? Here we see the difference. Now, when we recouple brown fat mitochondria from wild-type animals, we can start uncoupling at a much lower concentration. So the fatty acids, when UCP1 is present, additionally reactivate, and they overcome the inhibition caused by the nucleotide. So we think the situation within the brown adipocyte is that we have inhibitors that are normally active, and then we have activation by probably fatty acids or perhaps their derivatives, which starts then the thermogenic process. And these arise because we have control of the brown fat activity from the sympathetic nervous system, which is in its turn regulated from the hypothalamus by signals from cold or food, releasing norepinephrine to bind to beta-adrenergic receptors, mainly of the beta-3 type. This causes some lipolysis to provide some substrate and also then activation of the UCP1 to allow heat generation. When we then look at the cells, we can look at both those processes, both the activation by norepinephrine and by fatty acids. And let me show you this. Here in cells, isolated mature brown fat cells not containing UCP1, the knockouts, we then have a low rate of respiration, we add the sympathetic transmitter norepinephrine, and essentially nothing happens. There's no thermogenesis in the absence of UCP1. However, if we have wild-type cells containing UCP1, norepinephrine has a dramatically different effect, causing a large amount of thermogenesis and respiration. So UCP1 makes the brown fat cells thermogenic. And if we add fatty acids at a reasonable concentration, again to the brown adipocytes from the UCP1 knockout animals, nothing much happens. And then we do it in cells from the wild-type animals, and we see that oleate, in this case, mimics norepinephrine, and we get a dramatic increase in thermogenesis. So this is how we think the cells are working. But where do these cells come from? What are they? They look very nice, but think about the large amount of mitochondria I showed you in them. For many years we were interested in this phenomenon, and some years ago we made a global gene expression comparison between brown adipocytes that had been derived from, or precursors from brown adipose tissue, and white adipose tissue that we cultured under identical conditions and then performed global gene expression analysis. And what we found, one of the things we found when we made a table of genes that were enriched more than 5-fold in the undifferentiated pre-adipocytes, the brown versus the white, we found this list of genes here, and the ones I've highlighted in red, you'll note are myogenic genes. And so we've postulated, and this was later confirmed in elegant lineage tracing studies from Bruce Spiegelman's lab, that the brown adipocytes, the classical brown adipocytes and muscle cells, derive from the same MIF5 expressing progenitor cell, and that white adipocytes in general did not come from this lineage. So how did this happen? Well, of course we don't know, but if we think of the time when we were all fish, then we had large, if you think about dissecting a fish, we have here large epaxial muscles in the fish, and we also, these muscles derive from the dermomyotome, which expresses the transcription factor NGRAILD1. Now, in mammals, these muscles are not so noticeable if you think about it. We just have a few epaxial muscles. But what Atit showed was that also, not only these few muscles, but also the skin of this region and brown fat derived from the same NGRAILD1 expressing progenitor cells. So this is where brown fat, the classical brown adipocytes, seem to derive from. And we had this situation, which really is a little more complicated, and I have to now put in the bright adipocytes, UCP1 expressing cells, which seem to have another embryological origin, possibly a common precursor with white fat, although this is not clear, and there may be several of these different progenitor cells. But I'm talking really then today about all UCP1 containing cells. What did we gain as mammals through the development of UCP1 and brown adipose tissue? What did we get by not having heat production from brown fat to having heat production? Did this influence basal metabolism? Well, I showed you before the brown adipocytes stimulated by norepinephrine. What you probably didn't notice or think about was that, in fact, prior to stimulation, the basal respiration is identical. So there's no leakiness inherent in UCP1. And in fact, we see then that when we look at the metabolism in intact animals as a function of the amount of UCP1 in an animal, actually there's no influence whatsoever of how much UCP1 we have on basal metabolism, in agreement with the fact that UCP1 is not leaky. And this is maybe a caveat for our friends in the pharmaceutical industry, that just by making more brown fat, we're not going to change metabolism. Brown fat has to be activated if it's going to influence our energy expenditure. So what did we gain from developing brown adipose tissue? What we think primarily is that UCP1 facilitates our life in a cool environment. The Stockholm University campus last winter, our experiment here is that we're transferring mice that have been living, regular wild-type mice that have been living at 30 degrees and have a certain low basal respiration, to a cold environment, and they start to shiver dramatically and elevate their metabolism, their thermogenesis. In animals that lack UCP1, they have the same acute response to cold, but even after 3 weeks in the cold, they're still shivering just the same as they were when we first exposed them to cold. If we look at the wild-type mice containing UCP1, we see, as I said, the same acute response here. Immediately they start shivering. However, after 3 weeks in the cold, the shivering disappears. They no longer demonstrate shivering, but they still have the same high metabolism. Of course, they're still defending the same body temperature. They still have to be careful about this and produce heat. And this then is the paradigm of what can be called classical non-shivering thermogenesis, that with time in cold, shivering ceases. And we have then a development of mitochondria and more UCP1 in the tissue, and we have the possibility for heat production. But if we don't have this in knockout animals, of course, what can happen is that we can shiver, we can exercise. And so, in fact, what we see is that while cold acclimated animals of the wild type have elevated metabolism to defend body temperature, so do the UCP1 knockouts. They have the same level of thermogenesis, but now they're doing it by the more uncomfortable shivering. And so this is the benefit, as we see it, of having developed brown adipose tissue. It allows really a more comfortable life in the cold, so that mammals can, for example, develop a niche of being active at night. They survive better in cold, and it's much more comfortable, but it isn't an absolute necessity for survival. But it is more comfortable, but it does demand, of course, any increase in metabolism demands an increase in food consumption. So the animals have to have access to 2 or 3 or 4-fold more food than they did in the warm environment. Now, this is one strategy then for living in the cold, that you can maintain your body temperature if you have enough food to elevate your metabolism. However, there is an alternative strategy adopted by some mammals, and that is that they give up. They allow their body temperature to fall, and concomitantly, of course, metabolism falls, but then they have to be able to warm up, and they have to be able to do this by increasing their metabolism. And so what I'm talking about, of course, is a hibernation bout. And here we see data from the European hamster entering hibernation. This is just body temperature, maintaining during hibernation a low body temperature, marginally above environmental, and then arousing from the hibernation and increasing body temperature very rapidly. So we believe that brown adipose tissue is essential for arousal from hibernation, but we haven't been able to test this because there are no UCP1 knockouts in hibernating species, at least not yet. But we do see, this is data that is actually now 45 years old from Charlie Lyman's group, where he was working with bats. This is not an abbreviation for brown adipose tissue. This is the animal bat. And he's watching the animals arouse. This is their body temperature, and this is the brown fat temperature and the heart temperature. And you see how quickly the animal can warm up. He then tested by curarizing the animals. He gave them curare, and then, of course, they didn't feel very well. They had to be artificially ventilated. But once this had happened, then they were still able to arouse perfectly normally and in the normal period of time. So obviously, shivering, which was paralyzed here, was not a necessary requirement for arousal. So brown fat can do it. Whether brown fat is the only thing that does it in other animals is probably discussable, but certainly brown fat has a major role in this. And there's also a little story I want to include here about lipid metabolism in brown fat during hibernation. We were measuring triglyceride content in brown fat in animals that were adapted to the cold, and they were non-hibernating. They had a certain amount of total lipid. And then as soon as they went into hibernation, this lipid amount doubled. And then during the arousal period, over the few hours when they aroused, this was combusted back to the original level. And we calculated how much lipid they lost, how much would be needed to make this arousal possible. and it's almost enough. So that the total fat loss from the brown fat could almost arouse the whole hamster. There was one other little observation which I think is interesting and unexplained. We checked the different fatty acids and found that the fatty acid 20:3, homogamma-linolenic acid, was rapidly increased when the animal went into hibernation, and it disappeared immediately on the initiation of arousal before body temperature had increased many degrees. This rare fatty acid is a precursor for EPA, which gives rise then to prostaglandins, thromboxins and leukotrienes, but we don't know whether these are necessary components for the arousal process. And I think this is something which is fascinating but deserves more study at some point. So brown fat makes life in the cold more comfortable. Experimentally, we can measure brown fat activity through what we call norepinephrine-mediated or adaptive adrenergic thermogenesis. And let me show you what I mean by this. If we have an animal in a temperature-regulated chamber where we measure oxygen consumption and CO2 release, inject it with norepinephrine, then we can see the thermogenesis. This is an animal adapted to 30 degrees that is a knockout for the UCP1, and we get a small response to norepinephrine when the animal is injected. When the animal's adapted to 4 degrees, we get about the same increase in thermogenesis. So here, in animals that are lacking UCP1, there's no adaptive thermogenesis. The size of the response to norepinephrine is the same under all circumstances. However, if we have wild-type animals, we get, even in 30 degrees, a slightly larger response. And this is what is the really dramatic increase we see. We see this massive increase in norepinephrine response after the animal's been adapted to the cold. What we think is happening then is when we inject a bolus of norepinephrine, we're seeing this adaptive thermogenesis from the brown fat that's growing, but we also, of course, activate other cells. But these are the same cells that are activated whether the animal's been in the cold, whether it's in the warm, or whatever. So brown adipose tissue is the basis for the acquirement of the cold adaptation recruited adaptive thermogenesis, but not all adrenergic thermogenesis, because we do have some response in other cells. But this is non-adaptive, and I must stress this. What about the role of brown adipose tissue in the acquisition of diet-induced thermogenesis? Same type of response, same type of study, controlled diet. This is really what I showed you before. It's another series. Knockout animals, norepinephrine, small response. We give the animals, well, we could tempt them with something more interesting. In fact, what we actually give them is just a high-fat diet. Not quite as interesting, but they do enjoy it. But nothing happens in their norepinephrine response. After several weeks on a high-fat diet, they have the same size of response as they had when they were on the controlled diet. In the case of the UCP1 containing wild-type animals, the norepinephrine response, as I showed you before, is larger. And after the high-fat diet feeding, this is what expands, the magnitude of the norepinephrine response here, this part, over this background. And how is this possible? What's allowing this increase in norepinephrine? Well, this is then an increase in UCP1 that we can see here. We have, at a constant 30-degree temperature, a 4-fold increase in UCP1 on the high-fat diet. And this allows then for the increase in norepinephrine responsiveness. So we can divide up the responds to norepinephrine in the following components. Well, we have, of course, initially the basal metabolism, which is non-influenced by how much UCP1 or brown fat we have. We have then this bolus, this response to the bolus of norepinephrine, which we think is the pharmacological response of all cells, and this is independent of adaptation state. And then we have the true UCP1 dependent adaptive thermogenesis, which correlates completely with the amount of UCP1 that the animal possesses totally. So diet-induced thermogenesis is fully mediated, we believe, by brown adipose tissue. What's the effect of possessing this diet-induced thermogenesis? Here we have the fat weight of the animal measured by MRI over time, the UCP1 knockout animals, and wild-type animals. So a mouse with UCP1 finds it easier to control its body weight than the animal that doesn't have UCP1. After a high-fat diet, we have an increase in fat amount in the UCP1 knockouts, and in the wild-type animals, we have a slightly less increase. So this mouse with UCP1 is partially protected against diet-induced obesity, as long as they live at thermoneutrality. So why are they protected? Why do they have brown adipose tissue in this overnutrition state? Of course, we can't answer this, but mice are not normally provided with a large and interesting selection of foods. Their diets look much more like this, some acorns and some seeds and things. And the proposal from Mike Stock and Nancy Rothwell was that the reason why this mechanism has evolved is that the animals have to eat more calories than they really should. And this would be a disadvantage. They'd become obese and would more easily fall prey to an animal, a predator. If they have brown fat, they can get in the nutrition, the nutrients that they need without becoming as obese. Of course, we don't know, but this is possible. Before 2007, people always said in man, brown adipose tissue isn't interesting really because it's only found in newborns. But then through positron emission tomography, it started to become observed by radiologists using fluorodeoxyglucose uptake into tumors. They believed that they also saw uptake in other tissues. The fluorodeoxyglucose is taken up, phosphorylated, and then the positrons are emitted and collide with electrons. Energy is given off that can be measured, and people saw this type of uptake, symmetrical uptake, which was first termed tense muscle, and then in 2002 became realized from CT scanning that it was a fatty tissue, and eventually it was clear that actually there was active brown adipose tissue in a large number of adult humans. So it became accepted after 2007, and this has led to something of an explosion of interest in the tissue. Previously, it wasn't possible to use the fat-combusting property of brown fat to maintain or perhaps reduce body weight in adults, and we couldn't explain human metabolic phenomena as being due to brown fat. But now I think we can. There is this possibility to take this active tissue and use it to at least maintain or potentially reduce body weight, and maybe more importantly, to explain human metabolic phenomena as being due to brown fat activity. We can see brown fat here in these symmetrical sites in the neck, shoulders, and along the spinal cord. These patients are studied when they're fasted and in a slightly cool environment. So the glucose uptake dominates in brown fat. When we're inactive, we're not postprandial, and we have to be slightly cold. And if you think about it, this is probably about a third of our life when we're actually asleep. So maybe glucose uptake in brown fat could be important in a significant part of our lives. So for humans, does it matter whether we have brown fat or not? Does lack of brown fat make us obese? In the study with Saverio Cinti, these people who are in black here had brown fat and the others we didn't find on this occasion. This is the number, and correlated with the body mass index. So the slimmer people had brown fat. However, it also turned out if we plotted them as a function of age, there was also a tendency that it was the younger ones who also found. As we can see here, plotting BMI against age, these black dots are the brown fat containing people. So this is only a correlation, and one can ask, are people obese because they lack brown fat, or do they lack brown fat because they're obese and they're well insulated? Perhaps both's correct, I think. There's very little functional evidence at this point about the possible significance of brown fat, and there's just a tiny genetic evidence. There's a polymorphism in the distal enhancer of the UCP1 gene, and it turns out that if you happen to be fortunate and have AA at this allele point, then you have more UCP1 expression than if you happen to have GG. These people in another study, AA people, actually ate more than the GGs, and they had a higher energy expenditure. And they had a higher energy expenditure, particularly on a high-fat meal, compared with the GG group. And so they burn more energy after a meal, and this seems to be then brown fat dependent. And time goes, and it was then shown in a group of patients in the Czech Republic that the AAs tended more to be in the lowest quartile of body mass index than the GGs, who tended more to be in the higher. So there is a correlation here, but I say this is still not really explained. But the A's seem both to be able to eat more than the G's, and they have a tendency to stay more slim. Now, I showed you at first that norepinephrine binds to brown fat cells and activates lipolysis that reduces triglycerides by releasing fatty acids and activating UCP1. However, with time, and this is rather quickly in a cold environment, the triglyceride gets used up. And therefore, norepinephrine also causes synthesis of lipoprotein lipase to bring in circulating fat from chylomicrons. It causes synthesis of VEGF to make the blood vessels more permeable and allow for uptake of fat. And we know from earlier work from our lab that norepinephrine indeed stimulates lipoprotein lipase synthesis very markedly, whereas it inhibits it in white fat. So in experiments where triglycerides were given as a bolus to mice that were put in a cold environment acutely and had some brown fat, some German colleagues, Heeren and colleagues, looked to see how much of the circulating bolus of triglyceride was taken up by brown fat. And remarkably, brown fat cleared 50% of the bolus of triglyceride that was given. So brown fat seems to be able to counteract hypertriglyceridemia. Brown fat doesn't only use fatty acids for substrates, it can also use carbohydrate. And it can take up glucose under the influence of norepinephrine as well through a GLUT1 and through a GLUT4 pathway. And Heeren and colleagues also did the same with glucose. They had radioactive glucose, gavaged this and looked where it went. And here, 75% of the glucose they gave was disposed of in brown fat. This is pretty remarkable considering the size of the tissue, the weight of the tissue as a fraction of the total animal. So brown fat seems to be a major organ under these circumstances for glucose disposal and can could at least potentially counteract hyperglycemia and diabetes. So our mammalian prerogative to possess brown adipose tissue, its function, we believe, is to fight cold in a comfortable way and to counteract obesity and perhaps also then hypertriglyceridemia, hyperglycemia, which is the metabolic syndrome. Thank you for your attention.